Okay, so uh, this will be the outline of my presentation. I will start with a short introduction on the advantages of using DNA in establishing evolutionary relationships, followed by a brief overview on the methods employed in DNA sequence analysis. So, as I mentioned, I will be presenting the first part this morning, which is on blast analysis, and this will be followed by the second part of my lecture this afternoon on phylogenetic analysis. The objective of my presentation is to introduce online open source bioinformatics platforms commonly used in molecular phylogenetics. Mm. Nucleic acids are present in all domains of life, from prokaryotes to eukaryotes, including viruses. It carries genetic information and instruction that directs protein synthesis inside the cell. Nucleic acids are made up of the same building blocks called nucleotides, which include adenine, thymine, guanine, and cytosine. DNA sequence is the genetic material passed from parents to offspring or from parent cells to daughter cells. Thus, evolutionary relationship between organisms sharing similar characteristics can be traced by using this information. The nucleotide bases are essentially represented by only four characters: A, T, G, and C, which makes it very simple in terms of measuring or comparing the number of similar or different base composition in one DNA molecule between one species to another. Another advantage of using DNA sequence in reconstructing phylogenies is that it is consistent and not influenced by external factors which may arise from differences in the evaluation of phenotypic characters. In order to gain access or use the rich information encoded in the DNA, DNA is extracted from the organism under study and is sequenced. These techniques are becoming more inexpensive and can be performed in a very short period of time. The most important part in DNA sequence analysis is the assembly of the DNA sequence. After DNA sequencing, the DNA chromatogram, which shows the exact sequence of nucleotide bases in a DNA fragment, must be properly evaluated and has to undergo thorough proofreading. Short DNA fragments are manually assembled and should only include high-quality reads based on the chromatogram results. Normally, DNA sequencing companies will provide forward and reverse DNA sequences of the sample separately. These sequences should be quality trimmed in order to eliminate low-quality base reads prior to its assembly. Any single base change or erroneous base reads included in the final assembled DNA sequence can significantly alter the result of the downstream analysis, such as blast analysis and phylogenetic analysis. After DNA sequence proofreading and assembly, The final quality trimmed DNA sequence can be used for annotation using BLAST homology search and phylogenetic analysis. All DNA sequence data are stored in genetic sequence databanks that is easily and freely accessible for every researchers worldwide. GenBank, DNA Databank of Japan, and European Nucleotide Archive are the three DNA databanks where nucleotide and protein sequences are stored and all information are readily accessible through the internet. The three databanks are unified under the International Nucleotide Sequence Database. For instance, DNA sequence submitted and stored in GenBank is also accessible through DDBJ and EMBL websites. Now I will start my presentation on the first part, which is on BLAST analysis. This will include a brief introduction on the BLAST software and its primary use in DNA sequence analysis. This will be followed by a demonstration on its usage, and I will highlight important points in the interpretation of BLAST results. So Basic Local Alignment Search Tool, or BLAST, is used to perform homology search between DNA and protein sequences. BLAST can rapidly match a query DNA or protein sequence from all reported sequences in DNA databanks. BLAST is a computer algorithm available for use online at the National Center for Biotechnology Information, or NCBI, website. This bioinformatics pipeline has connected biology, genetics, and computer science to further advance our knowledge in molecular analysis of DNA sequence and protein sequence. There are 5 BLAST algorithms which require specific query data to be used in a specific database. BLAST-N is used for analyzing nucleotide sequences and uses nucleotide database. BLAST-P is used for analyzing protein sequences and uses protein database. BLAST-X, T-BLAST-N, and T-BLAST-X are variations of BLAST analysis which involves translation of nucleotide sequences and uses translated nucleotide database. These variations factors in the 6 spring translations in comparing translated nucleotide query. This is commonly used in the level of protein sequence homology search. BLAST software is very easy to use. It involves copying and pasting of the DNA or protein sequence or sequence identifier into the query text box, and the BLAST algorithm will perform the matching of every single characters in the query to all the reported sequences in the corresponding database. After which, BLAST results can be evaluated based on the coverage, quality of the match, and similarity of the query to the homologous sequences provided by the BLAST results. Okay, so for the demonstration, I will be using three common query data to demonstrate how to perform BLAST analysis. These include a DNA sequence. As you can see, it includes nucleotide bases, the A, T, G, and C. And we also have an accession number, a sequence identifier. So accession number is a newly reformed addition to the DNA data banks because it harmonizes the records of all the three data banks. And we're also going to use protein sequence with amino acid sequences in a single letter abbreviation. So I will start first in using the DNA sequence as my query. Okay, so you can just type BLAST in the Google search engine, and you can use it freely anytime using the internet. So you can see this program where there is a text box. It says enter query sequence, where we are going to paste the the nucleotide sequence that will be analyzed. Okay, so once pasted in this text box, so you can use the default settings. So it says database nucleotide collection and program selection, highly similar sequences. You can just click BLAST. So that's how simple it is. So once you click BLAST, it will then do the matching of the bases with all the other reported DNA sequences in the data banks. So this will be the result of the BLAST search. So if you're going to look at the result individually, the first thing that you should look at is the query coverage, which is the percent query sequence aligned to a sequence in GenBank or to a DNA data bank. And you should also have to look at the e-value or the expect value. So the lower the e-value, the higher the significance, meaning the alignment is highly significant. So usually the first item on the list has the best score. So as you can see here, the first on the topmost list is Pseudomonas Stutzeri Hero 3 with 100% query, an e-value of 0.0, and a percent identity of 100%. Percent identity is the similarity of the sequence to the coverage. Okay. So after checking these three parameters, the query coverage, e-value, and percent identity, then you can have an idea of the identity of the DNA sequence that we are analyzing. So most probably, the DNA sequence that we use is from Pseudomonas Stutzeri Hero 3, based on these characters, having 100% query coverage, e-value of 0.0, and percent identity of 100%. So below this organism, it has lower percent identity, so lesser the match, lesser the similarity. Okay, so for example, there are really close match. Like for example, here you have uncultured bacterium with 100 and a 100% coverage and 99.6% identity, you have to look for the uh, reference sequence, which is a complete genome of a particular bacteria in the same genus. So, for instance, below that second organism, we have a complete genome of Pseudomonas istsetseri. So, the genus and species is similar to the first one with the highest homology. So, most probably, the first one is really a Pseudomonas istsetseri based on the presence of this reference sequence uh, on the BLAST result. Okay, so once you have decided that, okay, so it's Pseudomonas istsetseri, so the topmost organism is probably the identity of the DNA sequence being analyzed, you have to check the accession number. So accession number, you can click on that accession number. It's underlined, and once you click on the accession number, it will bring you to the informations about the DNA sequence. So every uh, DNA sequence or protein sequence being submitted in a DNA data bank uh, are curated. Somehow they are peer reviewed. The informations, like for example, the authors, the source organism, the type of the molecule, the isolation are reported. So these informations are highly important when analyzing or using BLAST analysis. So you always have to look at the accession number. You just can click on it and uh, go through the informations provided. So another thing that you can do in BLAST analysis is there's FASTA on the top. Let's go back. There's FASTA below the name of the organism. So you can click on the FASTA, and it will provide uh, the sequence of the nucleotide or protein of the organism under study. So usually the FASTA format is used in phylogenetic analysis. So we are going to use this format later this afternoon. Okay, so the other variation that uses nucleotide sequence in the BLAST software is the BLASTX. So as I mentioned earlier, in BLASTX, the nucleotide sequences as query are first translated into amino acid, and then it uses a protein database to look for the homologous sequences. So it takes into consideration the 6 frames of translation of the amino acid. So let's see what will happen. So this nucleotide sequence is the same as what we have used earlier. So what will happen if we use BLASTX instead of BLASTN? Okay, so this will be the result. So again, we have to check the query coverage, the E value, and the percent identity. So we can see here that the query coverage is 51, E value is very high, and the percent identity is also very low. So most probably, this is not the identity of the nucleotide base that we are looking for. So the higher the E value, the lower the significance. Okay, so if you can also check its identity, the informations about the first one on the top list is hypothetical protein. So if you click on the accession number, so this is the information. So it shows the the amino acid sequence instead of the nucleotide sequence, which we obtained using the BLASTP. Okay, so now we are going to use an accession number instead of a DNA sequence. So accession number, you can find accession number in all publications, in journals, which deals with molecular DNA sequences or protein sequences. So accession number, you can use that in the 3 DNA data banks. So you can, if the sequence is reported in GenBank, you can use the accession number in the DNA data bank of Japan or in EMBL. So they are unified. And the accession number harmonizes the record of all the DNA sequence reported. Okay, so if I wanted to study a specific gene, I'll just look for a journal dealing with that gene, and then of course, once since it's already published, it already contains the accession number. So I'll just take note of the accession number and use BLAST to look for the information about the gene or protein maybe. So let's, for example, so going back to the BLAST software, BLAST site, you pasted or you have inputted the accession number in the text box, and you click on the BLAST N. Okay, so it provided you a result. Shewanella algae hero 1 gene for 16S ribosomal RNA. So again, we have to look at the query coverage, the e-value, and percent identity. So here, it's quite high. So 100% query coverage, 0 e-value, and 100% identity. So most probably, this is the identity of that accession number that we have used or we are interested to study. And then, as we go below the list, there's still some reference sequences, complete genome, with the same genus and species, Shewanella algae. Still, there are other several complete genomes. I just captured a fraction of the result. Again, we have to check the accession number. So click on the accession number. Okay, so here, so the information about the identity of that accession number that we have used and the complete DNA sequence. So, for example, you are also interested in amplifying that gene. You can now use this sequence provided in BLAST to design your primers. So that's the advantage. So if you want to just get the exact sequences, click on the FASTA again, and it will give you the FASTA format of the DNA sequence. So you can use that sequence to design the primers and use the design primers to amplify the same gene for your study. Okay, so not all accession numbers can be used in BLAST-N. There are accession numbers that are submitted with a protein or amino acid record. So for example, this accession number that I use, I entered in the text box, and then I use BLAST to analyze this accession number, and then it will give you a prompt. It says, failed to read BLAST query, GI, accession sequence mismatch, nucleotide input required, but protein provided. So it means that this accession number is not a nucleotide is not for nucleotide database. So once you have that prompt, you can just change the your parameters into for to match the accession number into a appropriate data bank, which is for protein. So if you click on BLASTP, which deals with uh, uh, protein data banks, then you will have the result. So using BLASTP, it says here the accession number that I provided with a protein record. It's probably a PAP2 superfamily protein. And we have to look again at the query coverage, the e-value, and percent identity. So it's very high, it's quite high. 100 coverage, 0 e-value, and 100% identity. So most probably this is the identity of that accession number. So we can also, again, click on the accession number and check for the information about the accession number. So, so it really has a protein sequence instead of a nucleotide sequence. So you can't really use BLASTN in analyzing the identity of that accession number in this case. But BLAST is very helpful because it will provide you a note that okay, so you need to change the parameters. You should probably use another database for your uh, BLAST search. Okay, so now we are on the last a type of information that is used for BLAST analysis, which is a protein sequence. So the usual protein sequence is in the single letter abbreviation of amino acid, as you can see here. So I'm just going to copy this amino acid sequence and again paste in the text box in the BLAST search. So uh, if you click on BLASTN, again, it's not going to match with the proper database. So when using a protein sequence, you have to use a protein data bank. So instead of BLASTN, you have to use BLASTP. Okay, so just change your parameter to BLASTP, and then after changing, usually the default settings are used. So it's for BLASTP non-redundant protein sequences, and so for program selection, BLASTP protein to protein BLAST. So the protein sequence will be analyzed using the protein data banks in the protein data banks. So so that this is the result of the BLASTP. So there is only one homology based on that amino acid sequence that we use. So this again, this amino acid sequence resulted into one or single identity, which is a PAP2 superfamily protein. And again, we have to look at the query coverage, e-value, and percent identity. So as you can see, the query coverage and percent identity is below 60%, and the e-value is quite high. So again, so standard operating procedure, we have to check on the accession number to look for to look for the information about this protein. So okay, so these are the informations about the probable identity of the amino acid sequence that uh, we are interested in. Okay, so uh, but for example, uh, that amino acid sequence you obtain then from a publication which states that okay, so this sequence of amino acid is a protein coding gene. It is responsible for the production of this enzyme or this metabolite that can be used into a specific application. So, in order to search or provide or get more information about protein coding genes, instead of using BLASTP, which is a more generalized homology search on protein databases, you can use the variation, which is a T-BLASTN. It takes into consideration the translation of amino acid sequences. So usually T-BLASTN is used to annotate protein coding sequences. So using the same amino acid sequence that we used earlier, now we are instead of using BLAST, I'm now going to use a T-BLASTN. So to investigate whether it is really a protein coding gene. So, so this is the result of the T-BLASTN. So it says it's a bacterium related to telluride, a reduction. So again, looking at the query coverage, the e-value and percent identity compared to the result using BLAST, the query coverage here is much more higher. The e-value is much more smaller, and the percent identity is higher. And again, we have to look at the information about this uh, data by clicking on the accession number. Right. So, so here, these are the informations about the protein coding gene, and as you can see, uh, it was submitted with nucleotide sequence instead of amino acid sequence. So, for me, if I want to study a protein and amplify that protein uh, in my organism, it's more helpful to get the nucleotide sequence where I can design my primers for PCR analysis. So, and so going back, since this the parameters that we are looking for, higher query coverage, lower e-value, and higher person identity, is much more favorable in this data, then most probably uh, I will use this information instead of the one presented in the BLASTP uh, result. Okay, so so after all our analysis, these are the BLAST annotation results. So using the DNA sequences, the query that we use is a nucleotide sequences DNA bases. So annotation is most probably it is Pseudomonas stutzeri strain hero 3 for the accession number query. So the most probable identity is Shawonella algae strain hero 1. And for the protein sequence as a query, so the most probable identity is the uncultured bacterium A1 DNA related to telluride reduction and tellurium formation. So that's how simple, how to do BLAST analysis. Okay, so so how can I use BLAST analysis in my research? So if you're dealing with, if you are interested in discovering proteins, enzymes uh, with industrial, pharmaceutical, or environmental applications, uh, uh, you can use BLAST as the first uh, method of analysis for your research. So this example is uh, more particular to bacterial cells because it's much more easier to culture bacteria and to collect bacterial cells. So if you're interested in discovering new enzymes or bioactive compounds from bacteria, you can just collect or first culture the bacteria, showing the activity that you're interested in, collect the bacterial cells in microcentrifuge tube, and you can store it in 40% to 50% glycerol. So instead of for example, the bacteria, the organism under study is still unknown, and you want to know its identity without going through the laborious process of primer design, DNA extraction, PCR, electrophoresis, and all. You can actually uh, send the bacterial cells stored in glycerol to a DNA sequencing facility. So so you have to communicate with the DNA sequencing facility if they will accept that sample. But in molecular biology, bacterial cells can be used right away in PCR reactions, and then because in sequencing facility they have to amplify the DNA first before sequencing. So the DNA cells that you have collected can serve as the template. So it's called colony PCR. So they will just use the cells directly into the PCR reaction, and then afterwards they can do the sequencing without DNA extraction. So there are fortunately we have now Philippine Genome Center that provides DNA sequencing services. So other DNA sequencing facilities that we have near the Philippines, in Singapore, there is a first base sequencing company, Solgen in Korea, or Eurofins in Japan. So, so you can choose whichever is comfortable. But of course, we we now have a sequencing company in the country, so why not utilize our own facility? So after if they accepted the bacterial cells that you have without extracting the DNA, then uh, better because you can save time and money in processing or identifying your sample. So after getting the DNA sequence from the DNA sequencing facility, then so you have to do the proofreading and assembly of the DNA sequences. And after getting high quality DNA sequence, you can do BLAST. So those BLAST results that you have is an information already that is publishable. So that's a shortcut method in using blast analysis in research, especially now during the pandemic. So uh, the mobility and accessibility of laboratories is hampered. So you can resort to this methodology. Okay, so uh, uh, in the Philippine Genome Center, this is you can see this on their website. The cheapest pricing on DNA
Nuggets.
using Sanger sequencing, it costs only 250 pesos per sample. So I think that's very cheap. But um, per experience, not all sequence results uh, can be analyzed. So it depends on the quality of the sample. So mostly, you really have to do the DNA extraction, the PCR part. But I have an experience that we only use the bacterial cells. We submitted that actually sa Solgen in Korea. We submitted our sample to Korea, and then we get good quality sequences. They have sequenced the 16S ribosomal RNA on our sample using the bacterial cells without going through the DNA extraction PCR on our part. Okay, so for the summary, so DNA sequence analysis using BLAST is very simple. Uh, you just always have to check on the accession number to get information about the data, and you also have to take into consideration the consistency of your query and the database that you're going to use. But the BLAST software can prompt you if you're using the right setting right away. BLAST software is an open source, meaning it's free and it is user friendly. It's very easy to use. It's a copy paste method. So if you have a DNA sequence from a journal, you just can copy that, or an accession number, you just can copy that, or an amino acid sequence, you just can copy that and paste on the BLAST text box and click on the BLAST search, and it will readily provide you the probable identity of your sample. So it is fast and easy. Usually it takes less than 5 minutes to get the result. And you can access the information stored in DNA databanks anywhere in the world. So, so that's all for my for the first part of my lecture. So, if you have any questions, maybe uh, we can entertain it before we end this first part of the lecture.